Okay, so a little bit of an overview over what I'm going to talk about today. I'm going to talk to you a little bit about our laboratory, talk to you about just briefly about some of the technology that we use, and then I'm going to talk to you about our acute human feeding model that we use as our main experimental platform. And I'm going to talk to you a little bit about how we got to the model we're actually using, and then how we actually are using it for preclinical testing. And then I'll show just a little bit of research that we've done to date looking at nutritional countermeasures. And we actually have about 20 ongoing projects right now looking at a variety of nutritional ingredients. And I'm going to show a little bit of some of that work today. And then certainly if you have questions about any of the other things we're working on, I'll be around all day. I'm excited to see all the excellent posters. I've seen a few of them up already. So I'm looking forward to interacting and connecting with you in that event. But if you have questions about any of this work, happy to address those. So our lab is located at the University of North Texas, which is in Denton, Texas, which is a suburb of Dallas. It used to be a distant suburb of Dallas, but Dallas has gotten so big that it's basically just on the edge of Dallas now. But we use a lot of technologies in the lab. The university is a little bit unique in the sense that we don't have core facilities. So that basically means that within the lab I work in, that I share with multiple investigators, we have protein detection facilities, we have flow cytometry, we have a genomics unit, and a few other units within the same lab. And so the main platforms we use for our biomarker testing is the LX200 and the MagPix. So just the next thing is just I want to kind of walk you through the overarching approach to the experimental model that we use. And so generally what we look at is we look at things and factors that you could consider lifestyle factors. So this includes things like dietary habits and more specifically poor dietary habits, which I'll show you a little bit of how we manipulate that in a moment. We look at physical inactivity and other factors within the lifestyle that contribute to disease risk. We then take these factors and we look at how they affect biological outcomes. So our chief biological outcomes that we look at is, and that we've gotten very interested in, is dietary endotoxemia. And I'll talk a little bit about dietary endotoxemia or metabolic endotoxemia in a moment if you haven't heard that terminology before, and how we look at its link to disease risk. We've done a lot of work over the years looking at weight gain and obesity and subsequent weight loss. We've done work recently looking at weight cycling and a few other factors. And then really these things all contribute to an overall increase in systemic inflammation, which is the main thing that we're usually trying to prevent. And then we look at how these biological factors feed into cellular changes. So how does that affect your pro-inflammatory monocytes? How does that affect the generation of modified LDL molecules? And how does that eventually result in the formation of foam cells? So there's a lot of stuff on this slide. Just to kind of focus this presentation, since I don't have a lot of time with you today, I'm only going to focus on a few of these effects today. So namely, the reason we're interested in all of these things is because a culmination of all these things contribute to atherosclerosis risk and other chronic diseases. So what we're really going to focus on today is how dietary habits influence these responses. I'm going to talk a little bit about dietary or metabolic endotoxemia, which I think is a really interesting and exciting area of research because I said so. And we're also going to look at systemic inflammation. So then this is the point where it's worth kind of walking you through the feeding model that we normally use. And normally the question I get at this point is somebody saying, how did you come to that? Well, we came to this model because when I started out, we used rodent models for our obesity models. And we used the classic genetic OB-OB model. We also did a lot of work with wild-type mice and manipulating macronutrient content in the diet, causing weight gain at different rates. And we also developed a non-lethal blood collection technique on those animals so that on a weekly basis, we could track changes in biomarkers using Luminex technology and then track them directly with changes in body composition. And what we found is that there were certainly some effects, some disease risk changes, some cytokine changes that did take 8 to 12 weeks to manifest. But there's other changes that we began to see in as little as 1 week of weight gain or a dietary manipulation. So that made us wonder how quickly some of these changes actually occurred. And it led us to speculate, and others have speculated as well, that actually the accumulation of chronic disease doesn't just magically happen after you've been unhealthy for a long period of time, but rather it occurs as a result of the accumulation of this daily change based on dietary habits or other lifestyle habits. So we actually developed this subsequent human feeding model that we now use. We validate it and we use it extensively to look at disease risk changes. And so I'll show you this model in just a moment. And then I'll show you some of the work we've done classifying the model and characterizing it with respect to biomarkers. So just to kind of walk you through with a diagram, because I love diagrams. So I'm gonna give you a really good diagram here and kind of give you an overview of what we know to date based on research from our lab and just from the field in general. So really the principle and the response that we're looking at is this dietary endotoxin response. Okay, so if you're not familiar with what this is, in a subset of the population, and actually we have pretty good data now to know what percentage that response is, an individual, if you feed them a meal which has a high calorie density and a high fat content, it interacts negatively with the gut bacteria, resulting in the death of some of the bacteria in the gut. And then they subsequently release lipopolysaccharide, which we characterize as an endotoxin response. Now, the good thing is in most people, when that happens, nothing happens. You don't get any endotoxin into your bloodstream. But in a subset of individuals who have issues with gastrointestinal permeability, you can actually have leakage of endotoxin from the gastrointestinal tract into the body. And you can usually measure this as somewhere between a 5- and 6-fold increase in endotoxin in the serum within 5 hours after eating a specific type of meal. And I'll show you that specific type of meal in a moment. And I think they usually have me present first because when I show you the type of meal, then you don't usually want to eat for a while. I can tell you though, I went over and I had breakfast and I approve the breakfast. So I approve of the breakfast. So we're good there, and I haven't seen what lunch is going to be, but I think we'll be good there too. So the bottom line is, in some individuals, they have this dietary endotoxin response. Endotoxin is particularly bad when it accumulates in the bloodstream because it can actually cause damage to the endothelial wall and cause subsequent release of endothelial microparticles into circulation. If you do flow cytometry, these are fairly easy to measure using common CD markers that mark endothelial cells and looking at particle size. The other thing that happens is endotoxin actually feeds back into adipose tissue, and it can also cause adipose tissue to increase the release of factors such as MCP1, inflammatory cytokines, and endocan-1. And all of these factors can then interact with monocytes in circulation and cause an increase of flux or transmigration of pro-inflammatory monocytes from the bloodstream into adipose tissue, causing subsequent adipose tissue inflammation and additional release of these same factors that then feed back into the liver and initiate an acute phase response, which it actually causes a change in a lot of different factors, but the ones that we see most often with this model are C-reactive protein and haptoglobin. And we can actually get transient changes in about a 5-hour window after this type of meal in haptoglobin and CRP. And then those acute phase markers interact with the pro-inflammatory monocytes in combination with aggregates that you get forming between platelets and EMPs. And essentially what ends up happening is you create anchor sites for pro-inflammatory monocytes in circulation. And that in combination with an increase in triglycerides, which also happens with this type of meal, you get an increased formation of modified LDLs, namely oxidized LDL. And as many of you know, that in combination with inflammatory monocytes creates the perfect situation for the formation of foam cells. And we've shown, not necessarily in this short-term model, we haven't shown an increase in foam cells, We don't get a lot of human participants that will let us remove their blood vessels during testing, so we haven't tested that out yet. But we actually have a really nice model where we can take out monocytes and we can test their capacity to become foam cells. And we can show within that 5-hour window there's an increase in propensity of monocytes to become foam cells when exposed to modified LDLs. So this is the response that we can actually generate, and we've generated it and replicated it using different types of high-fat feeding. And so just to quickly walk you through how this works, basically what we do is we have people come to the lab, and then we take a pre-meal blood sample. We then have them eat our special meal, and then we take subsequent blood samples during a 5-hour postprandial window where they consume no additional food. And normally the meal composition we found that is the most effective for what we're trying to accomplish is a meal where you get about 65% of the daily caloric needs and roughly 87% of your daily fat allowance. So this translates for a normal size individual to a meal that's roughly 1,400 kilocalories and roughly 75 grams of fat. So normally at this point somebody will say, well, it must be pretty hard to get a meal that can do that. Not hard at all, actually. Some of the things that we've used in the past, commercially available breakfast sandwich and hash browns, you will easily get to that number without much trouble. In fact, the hash browns are worse for you than the breakfast sandwiches. The other popular one we use from time to time is the burger and French fries. Again, pretty darn easy to get to that number. Usually we have to end up cutting back the portion so that we don't exceed that number. And then our current study model that we're using right now that the students on campus really love is cheese pizza. And so what we found is that in all of the individuals we've tested, and we've run close to 500 people through this model now, we find that roughly 45% of the individuals that we feed this type of meal to exhibit this dietary endotoxin response. And the ability to identify people that have this response is really important, in particular if you're trying to test a nutraceutical treatment to prevent dietary endotoxemia. If you don't enroll subjects that have dietary endotoxemia, then you find nothing. And if you look, in particular, I'll show some data we've done with probiotics recently. But one of the major inconsistencies with probiotic research at this time is, well, one is that a lot of probiotics don't really work. So that's a slight problem. But the other problem is that some of these studies don't enroll a patient population that actually has the response you're trying to treat to begin with. So we'll talk about that in a moment. So just to show some validation of this meal response model, one of the first studies that we did, and we've replicated it several times, is we wanted to demonstrate whether or not there were changes in these biomarkers on an acute basis, or whether or not those responses carried over into subsequent day. So in this particular study, what we did is we enrolled, I think there was about 25 women in this study, and apparently healthy, normal body weight, not obese, not overweight. And we had them come to the lab. We hooked them up to an IV. We fed them the meal. We took blood samples for the 5-hour window. And then we basically sent them on their way. We gave them a controlled meal for the rest of the day that was a low-fat meal to just fill out their calories for the rest of the day. And then we had them come back in the morning, repeat the exact same thing again. And we measured a variety of biomarkers, but if you look at the slide, you can see some of traditional, pushing the wrong button. You can see we measured some of the traditional markers, so cholesterol, triglycerides, glucose. And what these rings on here show is the dotted blue line is the response on the first day, and the red line is the response on the second day. And it's basically looking at a fold change over all the samples. And what you see is some of these traditional risk factors, you get a response that's really similar whether you eat that on the first day and then come back and eat it on the second day. But we found a few biomarkers here, Endocan-1, CXCL6, and CXCL16, which actually were enhanced and greater on the subsequent day. So this kind of leads to that idea that some of these changes in disease risk that we see over time are actually an accumulation of effects over multiple days. So then, of course, being curious as we are, we repeated this experiment and we basically measured even more things. And we started to look at some of our other factors, and that's when we went in. And the easiest way, there's no real easy way to demonstrate responses when you have hundreds of variables. Actually, one of our other speakers later today, as I was finding out before we started, has some really good data that they're going to show, a really good model for kind of consolidating some of this massive data sets down into something that's meaningful. But this is one method that we use to look at that. And generally what you can see if you look at this response, oops, God, I keep hitting the wrong button. What you see is that, again, comparing day 1 and day 2 response, each of these boxes represents a subject. So this first box here and this, if I can find the right box, that box, are the same subject. So basically what we did is we looked at a variety of factors and just kind of screened for which of those factors were stable day to day and which of those factors were enhanced. And some of the ones down here that were enhanced on the second day are definitely ones that you don't want to be enhanced on the second day. So again, looking at this effect and this role of pro-inflammatory monocytes, their response seems to be enhanced on a second consecutive day of eating this type of meal. And the thing that's really interesting is this isn't breakfast and lunch. This is breakfast and then 24 hours later, another breakfast. So these are pretty long-lasting effects. And so we've used this model and gone in and shown that most of the peaks of these responses is about 4 to 5 hours after the meal. And so you can see if a person's lifestyle, if they had poor or low nutritional quality for all their meals during the day, they'd eat breakfast, and then about the time they'd eat lunch, they'd have the peak response, and then they potentially would have a higher response than that, and then they'd eat dinner and have an even higher response than that. So over time, you would basically have this accumulation of changes that may predispose them to chronic disease. So obviously, there's a couple things that we learned from all this, is that daily dietary habits do matter. They absolutely do matter for some aspects of disease risk. So that led us to say, what can we do about that? And so it seems really simple. The obesity and most chronic diseases are preventable. If you have a healthy, active lifestyle, you eat a healthy diet, they're preventable for the most part. But the problem is that a lot of people are not particularly good at eating healthy and being physically active. And in fact, if you survey people about their dietary habits and their physical activity habits, there's no way we should have an obesity problem or chronic disease problem in this country, because everyone apparently does massive amounts of exercise and they barely eat. And if you look at survey data, that's what it will consistently tell you. Well, we obviously, I think, joking aside, we know that's not the case. So our work has focused on trying to identify ways we can manipulate the diet and potentially provide some protection against some of these effects without necessarily requiring someone to make massive changes in what they're doing. So this almost ends up sounding like an infomercial where you can lose weight without the hassles of diet and exercise. But that's not actually what it is. So we do promote a healthy lifestyle. We just find ways to provide that extra help by manipulating the diet. So I'm going to show a little bit of work we've done with dietary polyphenols. The data I brought today is actually, I think it's fairly interesting data, especially considering the type of dietary polyphenol we used. The participants that were in this study, they really loved this study because it involved dark chocolate and eating that on a daily basis for science. The other thing that I'm going to talk to you a little bit about is some of the work we've been doing with probiotics, some validation work of the actual probiotic itself, but then talking about some of the outcomes we've observed so far. And then there's always other things you can look at. So we have some ongoing projects that are combining different types of prebiotics with the probiotics. So if you're not 100% sure what that is, so the probiotics are the bacteria itself that you're trying to supply, and the prebiotics are the food for the bacteria, or the food for the good bacteria. And we're always very good about saying we want people to have a healthy bacterial profile, yet I don't think anybody actually knows what that means. If you ask different people that are experts in the field, they'll tell you what they think that means, but I don't think we actually 100% know that yet. And then we have other nutraceuticals that we're also looking at. So we've actually gotten to the point where we do a lot of ingredient claim testing. So we've had the opportunity to test a lot of different things that people think will improve health. So like I said, this data here, this is from a study that we did a couple of years ago that was funded by the Hershey Center for Health and Nutrition. And the goal of this study was to develop a healthy delivery system for dark chocolate polyphenols. And the actual technical term for dark chocolate polyphenols is natural cocoa. And so we basically figured out a way to make a bar, which did not taste very good, that had the equivalent of the dark chocolate polyphenols of about 10 chocolate bars in one bar, but only had 70 calories. And so it was a very effective way to deliver a large amount of dark chocolate polyphenols without also delivering a large amount of calories. So the subjects in this study, we basically recruited women for this study that were without any known disease risk, and they were either normal weight, overweight, or obese. And that was confirmed not only by using BMI, but also by using a whole body DEXA scan. That is actually a pretty important piece that's often overlooked when designing studies, especially studies of this nature. You'll see investigators that solely use BMI to classify subjects. That's a really bad idea. In fact, it's a really bad idea because a lot of the individuals we screened for this study that were normal body weight by BMI, they had 40 to 50% body fat. So technically by the body fat classification, they were not normal weight, they were obese. So always a study should have a secondary measure. And then what we did is we had all the subjects complete both conditions in a random counterbalanced crossover design. And we looked at just changes in a variety of biomarkers. And the study actually had, I think it had close to 200 outcome measures in it. These are three of the ones that I thought were the most interesting and actually made the best case for what I'm trying to tell you today. So they're the ones I picked. But what we have here is we have Endocan 1 response here. We have the haptoglobin response, and we have a serum TNF-alpha response. And in this particular model, what we found is that compared to the placebo, again, same subjects, placebo and treatment, and there's no order effects or anything like that, we found that in general, the treatment lowered Endocan 1 in both the overweight and obese subjects, and it slightly lowered it in the normal weight subjects, but not much more than the placebo and not significantly more. And then where we started to find really interesting things was a significant reduction in haptoglobin, but only in the obese subjects. And then reductions in TNF-alpha in, for the most part, the overweight and the obese subjects. And so this study was interesting for a couple of reasons, because we didn't do any other kind of dietary control at all. We basically just told the people to go do whatever they normally do, just eat this every day. And so it's interesting, without really manipulating body weight, there was no body weight loss or gain during the study, no change in body composition, that we were able to basically lower these factors that would be reflective of inflammation or an acute phase response. The last piece I'll show you today is with our work with spore-based probiotics. And in this response, this is a serum endotoxin response. And if you look, if you look right here, this is the pre-supplementation in the placebo group and the treatment group. So classic endotoxin response, 5 to 6-fold increase in endotoxin. And then after a 30-day intervention, we were able to blunt that response down to about a 3-fold increase. So we didn't completely erase the response, but we reduced it. This graph here on the top shows triglyceride responses. So we also blunted the triglyceride response with treatment, and we lowered the MCP1 serum concentration with treatment. So to quickly summarize what we have, so going back to this beautiful diagram I created, what I've shown you is with the work that we've done and the work I showed you today, and there's a lot more of this. So if you want me to bore you to death with details, feel free to come and find me, and I'm quite certain I can do that very effectively, according to my students at least. What we've shown is that with some treatments, we can blunt endotoxemia, we can blunt some of these factors, we can blunt the potential for more modified LDL, and that should subsequently, we've demonstrated that we can reduce endotoxin, reduce EMPs, reduce aggregate formation, reduce monocyte adherence, and reduce the acute phase response. And then our ultimate goal in doing all of this is trying to reduce the potential to form foam cells. So I think I've shown you some to show that's possible. Obviously, there's always more work to be done. And just in closing here, and I think we're going to open the floor for questions in a moment, but I'd like to thank my collaborators in the lab and some of my students that have contributed to this over the years. I keep hoping my students will go into academia, but they keep being hired by the people that I buy stuff from. And then also to Luminex just for making this event possible and bringing me here from Texas and away from the 97-degree heat we're having right now. So thank you.